Good afternoon. My name is Gabe Hitchcock, and I'm the COO and co-founder of Onconetics Pharmaceuticals, where we are engineering the future of gene therapy. Now, as some of you may know, gene therapy is an approach wherein genetic material is added to a cell in order to catalyze a therapeutic effect. However, the problem with gene therapies developed to date is that they're not selective. What this means for a patient is if they're given a gene therapy, while it may get into the intended tissue, it often ends up in other parts of the body where it can cause serious harm. But what if we could create a therapy that would specifically target only diseased tissue and not healthy tissue? With over 16 million cancer patients in the US and hundreds of billions of dollars spent every year on treatment and care, the value of such a therapy is tremendous. At Onconetics, we were solving the problem of selectivity by engineering a gene therapy that only activates within the distinct regulatory environment of a cancer cell. We achieved this by using small, non-coding RNAs. These are RNAs that are only present within distinct cells in the body and act as a functional off switch for gene expression. When a messenger RNA is encoded in the cell, it floats around waiting to become a protein. However, if the corresponding SNC RNA is also present, then it binds to and prevents expression of that protein. This process is called RNA silencing, and it's happening all the time in every one of our cells and every one of our bodies. At Onconetics, we found a way to leverage this basic genetic mechanism to create a trigger for a kill gene that would only activate within cancer, not within healthy tissue. We call this approach genomics-informed drug design. We start by taking genetic information from cancer patients. We then use that information to create our trigger, which is incorporated into our gene therapy and given back to the patients. The first step in this process involves the identification of differentially expressed SNC RNAs. These are RNAs that are only found within distinct cell types in the body. Now, when we looked between cancer and healthy tissue, we found not 10 or 100, but thousands of these RNAs that we could choose from to guide our therapy. From these, we then find the highest differentially expressed and incorporate those back into our gene therapy as targets for SNC RNA. Constructed like this, if our therapy finds its way into healthy tissue, then the small non-coding RNAs that are present in the cell will bind to and prevent its expression. The added benefit that comes from the heightened selectivity of this platform is that it allows us to be delivery agnostic, which is a pretty nice place to be for a pharmaceutical company, because it means that we can be very flexible when it comes to choosing our delivery mechanism, because we're not dependent upon delivery to achieve selectivity. To date, we are partnered with Karma Biotechnologies, a tissue engineering company, to use their nanoparticle system to create a viable product. Now, when our therapy is introduced to a patient, one of two things can happen. In the case of a cancer cell, the DNA is taken up, it's identified as native, it's read, our kill gene is expressed, and the cancer cells die. If it's taken into a healthy tissue, then the small non-coding RNAs that are present in the cell will bind to and prevent expression of our kill gene, thus leaving the healthy tissue unharmed. And when we first came to IndieBio from UCSF, we wanted to validate this approach within breast cancer. And so we chose just one small non-coding RNA to guide the expression of our therapy. These are the results from that study. This is our healthy breast model. You'll notice within the first day there's a pretty sizable dip, which we attribute to some noise, not from the therapy itself. But the important takeaway from this graph is that during the course of the experiment, there's very little difference between the treated and untreated healthy breast cells. What this tells us is that the single non-coding RNA we chose was sufficient to prevent expression. Now compare this to our triple-negative breast cancer cells. And you'll see there's a pretty stark difference between treated and untreated. By the fourth day, we've killed about 50% of the cancer cells. By the 8th day, we've killed 90% of the cells. And on to the 9th, 10th, and 11th day, even when the cancer cells that are untreated begin to grow and proliferate, we continue to achieve selective killing and repression. Next, I'm going to show you two videos from this experiment. On the left side of the screen, you'll see our untreated cancer cells. On the right side, our treated cells. As expected, for the untreated cancer, there's very little change during the course of the experiment. However, for our treated cancer cells, you can see there's almost immediate death as the cells begin to flicker out. And by the 9th day, we've again killed nearly all of the cancer cells within this population. To date, our pipeline consists of our triple-negative breast cancer and pancreatic cancer. However, because what we're engineering here is a bioengineering platform for creating targeted therapies, as we move forward and expand our company, so too will our pipeline to include these additional indications. Onconetics owns full rights to its IP, has filed provisionals, and is working with Cooley LLP to strengthen and expand our portfolio. The founding team consists of myself, Gabe Hitchcock, my co-founder and CEO, Luke Grunert, who also invented this technology while he was at UCSF, and our chief scientific officer, Jeffrey Sargent, who has a storied career both in academia and in biotech. We've surrounded ourselves with an advisory board of leaders in the industry. They include Admiral Joxel Garcia, who's the former 13th U.S. Assistant Secretary of Health, the former Director of Cancer Prevention at MD Anderson, and the current Chief Medical Officer at American Express. As well as Dr. Michael Kmark, who's the former Director of Product Development at Pfizer and current CTO at Vir Bio, and Dr. Alexander Kam, who's the former Executive Director of Oncology at Amgen and the Global Head of Oncology at Novartis. Our scientific advisors include Drs. Rossi, Morris, and Walters, each of whom are world leaders in their respective fields of gene therapy and non-coding RNAs. At the present moment, we're in the midst of our in vitro validation, moving very quickly toward our in vivo animal studies. By 2020, we anticipate being pre-ND ready. Throughout this process, we'll continue to optimize to increase both the efficacy and the selectivity of our therapy, as well as expand our portfolio. To achieve these aims, we're raising a $5 million seed round, for which we've already received several soft commitments and much interest. My name is Gabe Hitchcock, and we are engineering the future of gene therapy. I invite you to join us. Thank you.